based on a 10-point process. So what you'll see is a number of earned points, for example, 28 out of 30, for example. So, and I didn't have time to do this, but go ahead and just calculate a percentage of that. So you have a percentage based on 20 out of 30, and that happens to be 72.5, and that's basically 7.05, right? So basically 7.1% of what you earn for exam or quiz number 1. Okay? So make sure you do the calculations because it is only worth 10 points. It's on our first quiz. So make sure you do that. So I just, the reason I broke it down like that is it's quantitative in terms of how I measure the amount of points you get for each, for what you say basically. Okay, so it's very calculated in terms of how that works. Okay, so let's go ahead and pass these out. Raise your hand when I say your name, or attempt to say your name. I may not pronounce it correctly, but I'll do my best. Leslie Coates. Coates? Okay, all right. Yeah, there might be first names and honors and stuff, so. Deanna Wilson? Derek? Wilson? Wilson? Okay. Cody? And Cody? How do you pronounce your last name? Mengedi. Mengedi? Yeah. Okay, Cody? Yeah. Okay, very good. Mengedi. All right. Marla Eichert? Rachel Willendorf? Corey, Kaylee Miller, Keeleen Oliphant, no Keeleen, right. Daniel Henton, okay. Michael James Jamali, okay. Please. Yes, sir. Is it Kenny? Yes, sir. Okay. Thank you. Morgan Youngblood. Here.
Thank you.
Nick Nelson. Marissa. Okay. Melissa Yang. Okay. Ashley Harris. Hunter Bright. Right here. Tiger. Moran Smith. Randy Johnson. Garrett Lentz. Chelsea Zine. Justin Galloway. Craig Lamb. Jenny Soto. Okay. Matt Smith. Leslie Harris. Sheldon Davis. Cassidy Efron. Sydney Green. Amberley. Rachel Fletcher. Jason Davis. Caitlin Fan. Taylor Chapman. Taylor Conley. Robert Salt. So there you go. So again, take a look at it. Remember, what you write is really important, and how you write it with clarity is really important. And so it's not about definitions. It's about your ability to communicate through writing to me to convince me that you understand what we're talking about. Okay? So that's the key. So there's two parts to learning this well in this course. Number one, you start out with just the basic definition of what we're talking about, right? Which means read the text, read the notes, listen to the lectures, feel comfortable and solid in terms of what they are, right? And then the second part is put some thought into it in terms of how can I now explain this to somebody, okay? And so that's where you have to really work on this. I would suggest, how you do this, you can either stand in front of the mirror and talk to yourself if you want. But I would suggest other ways. Talk to somebody else, explain to them. How do you do that? That's why I established groups. So what I'm hoping is that the groups we created somehow develop into outside mechanisms where you can get together as a group and go over and study for the exam. Okay? And so that's now your time to explain to your group members some of these topics. Okay, so let's, Dr. John says, let's see if we can do this. Explain to me explain me what the peptidoglycan structure is, important components. Probably a better question would be explain the synthesis process, right? Tell me how this is formed. And let's see how it goes. I mean, it'll either be very elementary and your group members won't have any idea what you're talking about, right? Because they're like, wow, I just have no idea. I have it all memorized, but I just can't translate it. So that's going to be the trick right there, okay? How you can do this, okay? And then beyond that is going to be thinking about how this applies to basically how it affects our lives, okay? We know that this is a bacteria structure. We know it has a lot to do with certain types of antimicrobials. We got into that. We talked about growth. We talked about biofilms and things like that. So how do you get that information? Again, the textbook is going to be really important. The 2 research articles that I gave you, the introduction, that is really helpful. And if you didn't read the paper, definitely read the paper. As I said, the introduction, because that's, as I said, it's like a textbook. It gives you a really good, clear idea, and it challenges you because it uses a scientific method of terms that typically scientists use, as well as students. And that includes yourself, to be able to understand what they're trying to say. Okay, so your ability to give me the answer based on that information has to be something similar. Okay, so the level in which they explain things in those introductions, the terms they use, I would expect you to be able to use those. Okay, so think about that. So that's the challenge right there, and it pushes you a little bit more than just simply saying, well, you know, piece of cake. All we got to know is peptidoglycan. I know what that's about. Penicillin, I know what that's about. Biofilms, got it. You know, that's probably not the ideal. That's probably below average grade. If you're satisfied with D's, then I guess that's what you're destined to get. If not, push yourself. You know, see if you can convince me in your writing that you know the material. You understand how to apply this information, right? That's the key. So we'll see how that goes. All right. So while I load up here, are there any questions? I can talk and work at the same time, so it's not a big deal. Take a look at your. You have any questions about the exam? Happy to ask now, after class, office hours, you know, always available by email. You know, just keep that in mind. Always willing to talk to you guys about that. All right.
Are we going to need to buy a blue book or anything?
No blue book. I will supply the hard copy. So what I do is I'll ask questions and I'll be specific about the amount of room that I would expect you need to write your answers. Okay. So if I give you half a page, that's your limit. If I give you a space like that, I'm really looking for a pretty quick answer. Okay. So that's kind of your guide. So yeah, the case and hard copy exam I give you is essentially what you'll have. You know, to answer questions. Now I don't want to alarm you. You know, sometimes people get alarmed. Oh no, you know, I'm really worried about this test. No, just relax. It'll be all right. You attend class and you read the material and you think about this, and you thought about it before anyway. Right? You're at the level now where you're seniors and juniors, so you're thinking about stuff and you have ideas. And that's the beauty about my exams is that I present a certain angle, but if you're familiar with another angle because you're more informative about something related to this, put that in your answer. That's a good answer. Okay? So it's not always about I've got to learn what he's giving me, and I've got, you know, don't go that far because then it puts too much stress on you. Go with what your strengths are, your interests, and then use that as a tool to help you learn the subject. That's the way to go. Okay? So don't panic and say, you know, oh no, I'm really gonna bomb this. It's not gonna work for me. No, it'll be all right. Don't worry. But I do want to, and the reason I say it is I do want to stress the seriousness in this at this level as far as learning the material. So that's why I really push that. It's not something that you should take too lightly and say, oh, you know, I can study for this and get it done in a few minutes, and that's that. It's really one that I think the students that enjoy learning, like the subject, put the time into it, and give them a really good answer, and do really well. And then the others that just sort of take the notes, and that's it. You know, you get a level of average grade, right? And then of course the ones that are struggling, those ones certainly I would seek some help. Myself would be part of that process of helping. Make sure we understand the material. Okay. But the key is, you know, push yourself and really try to get some really good information. That's a A grade, no doubt. And then anything less than that, it'll stand out. You'll know as we look at those exams more closely. Well, you understand, but you're not quite at the A level, right? You might be an above average student, maybe average. But then that's where you really learn. Maybe I ought to push myself a little bit more, or read a little more, think about it a little more, just go a little bit beyond where you might be comfortable with. Okay, so that's the thing. All right, so I'm going to pull up the review, get on the projector. and we'll go through this, and I'll kind of set up the plan. Because the outline is really the test. So really the test here, we'll stare at the test here. This is it. And so the introduction, and I'll go ahead and present it here, is how we're going to handle it. So if you look at microbiology and how we started this class, we started growth, but I think a good way to really put this in perspective in terms of historical events, we know bacteria play a role in disease, and everybody's interested in that. And so one of the early discoveries, as we said, in the 20s, was this discovery that a fungi produces something that causes bacteria, it inhibits the growth of bacteria. And it took 20 years for them to develop and discover this substance, this agent, and it was identified as penicillin, right? So in the 40s, they began to administer penicillin, and it was huge because that was right around World War II, where people who were wounded, who had different types of injuries, you typically died because when that became infected, you died. So the death rate prior to that was huge. And so this was a big deal. I mean, it saved a lot of people. Might even saved some of your grandparents. You think about it. If your parents know of somebody that went to World War II, survived the wound, thankfully they discovered this. Big deal, right? Means you're here. So anyway, so it's a big deal. So anyway, that's how we'll start it. And we'll say, okay. So as we think about penicillin, we need to begin by thinking about what's the target. And what they discovered was that not only was this the agent that actually caused bacteria to stop growing, it also was interesting to think about exactly what the mechanism is. So the question would be, what's the target? Where is this penicillin molecule? What is it attacking, right? What part of the cell? So they discovered that this structure, which is a major part of bacteria called peptidoglycan, is what played a key role. So as you begin to think about that, then we need to say, okay, well, what is peptidoglycan? So you need to know, the first question would be, let's think about what that is. Describe for me what this structure is, okay? So it's really important for you to know specifically the names of this molecule. So it's really two parts, right? Consists of two different sugars, right? And I'm going to give you this image on the exam. And it consists of a series of amino acids. I would prefer you call that a stem peptide, okay? Now I've included some of the terms that I had on your preliminary review exam, and I just copied them back here. I might have added a couple more, put them in the order where it makes more sense. But I would expect you to use some of these terms to describe the structure, okay? So you can't just say two sugars, you're going to have to say two sugars are N-acetylglucosamine and N-acetylmuramic acid, right? And then you're going to have to say amino acids make up this structure called the stem peptide, okay? And so you need to be able to begin to describe that. Now there's important parts of the structure. What's this beta-1,4 linkage? Remember, that's how the two sugars are connected. So what are the numbers designations? Make sure you understand that, right? Those are the carbon numbers. Okay, carbon number 1 links to carbon number 4 to the other sugar. And that's how you can communicate between each other in terms of how these two sugars come together. It's a beta-1,4 linkage, covalent bond. Okay, and so that's important. This lactyl group is another part of the structure, and it connects the stem peptide to the two sugars. Okay, it's really important. It's right here. And this whole thing comes off of only one of the sugars, right? The muramic acid. So, and that's what makes it unique. So each muramic acid has a stem peptide coming off of it. Okay, so that's important for understanding that links the stem peptide and the two sugars. Of course, we call that stem peptides. the stem peptide made of amino acids, right? Now, what's unique about that? We talked about this, particularly this. What are the L and the D designations? Those are what we call isoforms, right? If you remember your days in chemistry, it's like the hand, right? The mirror image. This is similar, but they're really different, right? Because if you overlay them, my thumb is sticking out this way, and that would be the D form, and this would be the L form, and they're not the same. So when enzymes bind to molecules, they recognize different orientations. Okay. And so if an enzyme only works for in nature, in our cells, they prefer the L form. And then sometimes in nature, if there's a D form in there, which is this one, your enzyme will recognize it because it looks different. Okay. And so that's really interesting when you think about it, because the forms in which molecules can exist really is one of the ways in which we can begin to use the word specificity. Okay? And your textbook talks about that, right? It says bacteria are resistant to certain kinds of enzymes that degrade the sugar. Why is that? It's because the two terminal amino acids on that stem peptide are a D form. In nature, most of the time, the enzymes that are involved in forming bonds prefer to have L forms. Okay? So you have a cell that has these different D forms, and obviously enzymes that are involved in breaking those bonds are not going to work. And that's why bacteria are so uniquely resistant to many kinds of digestive enzymes. Okay? They're the only ones that have these D forms. In our cells, we don't have these D forms. Absolutely not. So it's kind of unique that they do that. So that's why I would like you to understand that. In your textbook makes mention of that. So read that carefully. Okay. So now we know what they are. Okay. You can describe the structure for me, and it's important because we want to understand how the penicillin works, right? So then before we get that far, you need to answer the second question, and that's, okay, what's the synthesis process of peptidoglycan? How is it formed? Okay, you need to understand that, and you need to explain to me how this happens, because that's the key piece to explaining to me how this penicillin molecule works. That's the key. So not only do you need to know the structure, you need to know how it's formed. We call it synthesis. Okay, so these are some important aspects of synthesis as far as terms that you really need to know. Okay, starts off with there's a really nice image in our textbook that talks about the cytoplasmic side of the cell and the periplasmic side separated by a membrane, right? And so all the synthesis events happen in the cytoplasm first. Okay, and key things that I would like you to know is it begins with having a carrier. It's called the UDP. That stands for uridine diphosphate. These molecules are in cells, and they bind to other molecules, and they actually allow those molecules they carry them. That's why they call them carriers. They carry them to areas in which they can now engage in synthesis. Okay, so these UDP carriers are critical for binding one of the sugars, and as the sugar makes its way toward the periplasm, it undergoes synthesis. Okay, and so when the sugar begins to synthesize into peptidoglycan, it now begins to add, particularly the muramic acid, because that has the stem peptide, right? The synthesis process begins now. The carrier is pulling it along. It's now beginning to add one amino acid at a time, okay, building it into a pentapeptide. And when that forms, we call that the stem peptide, right? Now, the key piece that I want you to understand about that is, remember, when we talk about the formation of a series of amino acids, we call that a polymer. That process is really unique and different than another type of polymer formation that we use the term translation, right? And that's the formation of a protein. The protein is just simply an amino acid strand, right? Now, we talked about that, so I want you to understand the difference. Remember? What's the conservative aspect of translation versus stem peptide synthesis? They require a transfer RNA to deliver the amino acid. Okay? In this case, the stem peptide has a transfer RNA delivering an amino acid in which a specific enzyme creates the first bond. And then the second amino acid comes in that's attached to transfer RNA, and another enzyme creates that peptide bond, so on and so forth until you build that pentapeptide or stem peptide, right? That's all it needs. Okay? Now in translation, it does require transfer RNA to deliver the amino acid, but it requires a little bit more as well, right? It requires a message RNA, it requires a ribosome, okay? And when it delivers that, it now begins to build a series of amino acids linked together by peptide bonds, just like the stem peptide, right? And of course, it grows beyond the 5 amino acids. That's what proteins. Proteins can be 100, 200 amino acids in length, right? And then that's how proteins are made. So that's the difference. It's really unique. Your book gives an image of that, and it's the way it's described represents a little bit of some confusion, and Daniel brought this up. But I want to create some clarity, is that transfer RNAs are still there. What they say is transfer RNAs and ribosomes, yes, together are not required, but the key is the ribosomes are not. Okay? So the conservative part is transfer RNAs, instead type synthesis, and translation. The difference is translation, message, and ribosomes are required, whereas in stem peptide synthesis, ribosomes, message not required. Okay? So remember that. That's the key piece. Because remember, anytime that we begin to mention what's different about bacteria and what's different about our cells, that really becomes an example of how you can now develop drugs that can target those differences. Okay? So if this question is ever asked again, that's the key. That's the best kind of drug you could develop, is one that would attack a unique structure of bacteria only that you knew did not exist in our cells. Because obviously, if you had shared systems, then it would attack us as well, and that's not good. And hence, side effects, right? A lot of times you hear about it. That's part of what happens. Is that it doesn't target always the cell you're after, whether it's a cancer cell or whether it's a microbial cell. That affects normal cells, which are our cells. Bad things can happen. Okay. So it's really important to use this terminology to describe all aspects of that. So I mentioned in there a couple of key enzymes. Okay, we have to understand what a transglycosylase is. That links the two sugars. Okay, so as I said, one sugar is carried by the carrier. It builds a stem peptide, and now it links to the second sugar, and that's due to the transglycosylase enzyme. Okay. And so as it goes from a 1 sugar stem peptide to a 2 sugar, there's a stage description of how we define that. And that's what we call the lipid 1, lipid 2 stage. Make sure you know what that is. Lipid 1 is the 1 sugar stem peptide only, being carried. And as it now begins to engage in synthesis, it now is 2 different sugars, and it's then peptide. Now it's a lipid 2. So they can define the stages in which the synthesis events are occurring, right? So that's the difference. Make sure you know that. So now once this happens, everything's in the cytoplasm. Yes?
Do you count everything before the linkage of the stem peptide and NAM as lipid 1, or just
It's lipid 1. It's lipid 1. So what happens is you got the carrier binding to the sugar, and you got the stem peptide formation right there, snapshot. That's lipid 1. Okay, and so that whole begins to now assemble by attaching to the second one. Boom, lipid 2. Okay, so that's how we tell the difference. So now this lipid 2, this is in cytoplasm, has to flip over to the other side because that's where the peptidoglycan is, right? And that's what it's going to add to. to allow peptidoglycan to grow, because remember, you visualize this is the bacteria cells dividing, so now it's beginning to form a septation, and we talked about that septation being heavily identified as having a lot of peptidoglycan synthesis activity. So that's where it's happening, okay? So anyway, and plus cells grow to what they call elongate. So if something elongates, it's not the cell stretching, it's new peptidoglycan added to it, allowing it to grow. So they kind of grow, get longer, and then they pinch in, separate. So the growth and then eventual completion of the pinching, that requires synthesis, right? So that's kind of how you can visualize this, right? So you can dream about it. So wow, it's happening now. I can see it. So you got lipid 1, lipid 2. It's going to flip across. How's that going to happen? It requires that molecule that's inside the membrane called a bactophenol. Why is that in the membrane? Bactophenol has a really unique characteristic of having a hydrophobic character to it. Okay? And what that means is it's water avoiding. Inside of the membrane, that's an area where there's no water virtually. Okay, so if a molecule is hydrophobic, it prefers to be in an area where there's no water. Right? The cytoplasm is full of water. The other side of the membrane, the periplasm, is full of water. But between the membrane is basically water-free. So if this molecule is hydrophobic, then that's where it's going to position itself. And that's exactly what it is. It's a hydrophobic molecule that's always there. It's not wanting to come out because there's water out. And then what it does is it doesn't just exist there. We discovered it actually binds to the lipid 2, and it flips it across to the other side on the periplasmic side. Now that 2-sugar stem peptide lipid 2 structure now can now be added to the existing peptidoglycan, right? Okay. So now how does that happen? Well, if you think about it, if the existing peptidoglycan is complete, you've got to break those bonds and open it up, right? So then you have to have these bonds that we call autolysis. They break them open, allowing now the addition of new peptidoglycan structures, right? So that process begins to happen. So it's really important to understand and to be able to incorporate autolysis as well in this process. Did I put it in there? I don't think I put it in there.
Yeah, you did.
Okay. So, and also, once that happens, there's another event that happens with the stem peptide at this point. Okay, so you've got the new peptidoglycan sugars being added. Okay, and now what happens to the stem peptides? They form a structure at this level now called cross-bridging. Okay, so as these stem peptides are coming off, they now begin to connect to each other, and we call that cross-bridging. So that also is beginning to occur. As you're adding new peptidoglycan, you're beginning to form these cross-bridges. Okay. And there's lots of ways in which this can happen. And that's why we call it a variant. Your book describes it as a variant. What do you mean by variant? It means it varies. You know, what's pretty much conserved is the 5 amino acids. That can vary a little bit, particularly at the end. But then how you link them, the cross-link can also vary. It can be direct cross-link from stem peptide to stem peptide, or you can have a series of amino acids between them to make the connection. We call it an interbridge. And it gets really, really interesting in terms of the complexity. So we're not going to go there, but just understand that bacteria have a whole variety of ways in which they can create these cross-bridges. And that makes them different from one bacteria to another, or then one type of bacteria, different strains, as they differ. And ultimately, all of that has to do with how they can avoid and survive different conditions. So they have these unique abilities to do that. So they're really quite interesting. But so you got cross bridges that now are happening that rely on the combination of peptidoglycan forming bonds, right, and autolysis, which break them to allow the insertions of these to go in. Now I did want to go ahead and have you think about, in general, how this cross bridge happens. So if you count the stem peptides, The first one being number 1 position, the book calls it position, so we can call it 1, 2, 3, 4, and 5, right? What happens is it does form what we call a pentapeptide, a lot of amino acids. So as it begins to form the cross bridge, there's a process in which that terminal 5th amino acid is released, and now it becomes 4 amino acids. And what happens is that 4th position amino acid now forms a bond with the adjacent stem peptide at the 3rd position. And you'll see this in the literature. It's called a 3-4 linkage. What that means is the 3rd amino acid of the stem peptide, which typically is this mesodiaminopolemic acid, better known as DAP. And it can be other things. That 3rd position binds to the 4th amino acid. Okay, I want you to understand that your book talks about that and mentions it. Okay, because that's the critical mechanism that allows cross bridges to happen. Is that you release the 5th one, immediately the 4th binds to the 3rd, boom, you got a cross bridge, right? And that's the process of that. And so the importance of the 3rd amino acid in think about it is really important. Okay, and the importance of having a 5-pentapeptide, the release of the 5th is important. So you have to have some things in place for this to be successful. Yes?
Remember the book talked about one or the other happening in gram-negative or gram-positive bacteria? Do you want to differentiate between the two?
No, don't worry about that. That has to do with the variants, how different they are. As long as you understand that one of the ways in which bacteria display their differences at the level of the cell wall, if you think about this would be a really good way to explain it to me. Okay, because it's quite different. It can be quite different.
Are the pentapeptide sequences different for different bacteria, and that's what makes it unique?
That's one of the ways. There is some conservation, like the first one is typically always going to be the L-alanine. Okay. Where it begins to differ, is the 3rd one. Sometimes it's the 2nd one can vary. If you just follow the level of differences, this 3rd one can vary quite a bit, and then the last 2 can vary as well. Okay. But that first one is pretty conservative. Okay. But overall, yes, the answer is yes. You can have that sequence of that stem peptide can vary.
So it's the sequence that varies and
The cross bridging.
Okay.
Because that interbridge, right? That interbridge. Because sometimes if it's direct, then there's no interbridge, right? It's just stem peptide to stem peptide. But if there's an interbridge, yeah, that series of amino acids can be different from bacteria to bacteria or strain to strain. It really is interesting. Of course, if you're interested in all of this, I'll teach you still a cell wall class, right? So you can always, everybody's going to enroll now, right? I mean, all excited. Even the graduates are going to come back, right? Pretty excited, yes.
So what does DAP do again?
It's the 3rd amino acid. It's just the name that's common.
Do you want us to know how to spell?
Yeah, well, yeah.
Okay.
That's part of the maturity that you have now, right? You can say not just the 3rd amino acid. You're going to say this is what it is, commonly known as the meso. And go on from there. So, okay. All right. So that's what I expect you to know, and be able to explain to me in use those terms, be able to achieve that description of how does that happen. Okay. So we can buzz through the rest pretty quick. Mechanism of action, that's pretty simple, right? Has to do with the suicide effect, meaning the penicillin binding protein has to bind to something. What does it bind to? It binds to that terminal D-alanine. And why? Because the D-alanine has a structure that looks really similar to the penicillin beta-lactam ring. A square-looking ring, right? It's in your notes. That structure is similar. So an enzyme looks at that and says, which one should I bind to? It doesn't really know. So if you have a lot of penicillin in your system, then obviously that's where the enzyme is going to bind. And if it does, then that's it. It doesn't form a cross bridge because it's binding to a drug. And so it essentially stops cross bridging. And when that happens, you compromise the cell's stability, and ultimately those cells begin to lyse and they die. Growth stops, right? And so if your concentration is low or there's no antibiotics, penicillin binding proteins bind to the two terminal alanines, form these cross bridges that we talked about, and you've got successful growth, right? So that's why they work so well. So now the question is, how does resistance develop? And we talked about two ways. One of them is the bacteria happens to have a gene that produces an enzyme called beta-lactamase. Okay, not all of them will have this capability, but if they do, that enzyme called beta-lactamase will cleave the penicillin beta-lactam ring. And when you do that, you disrupt its effect. Okay, so now the penicillin binding protein will not recognize that because the ring's been cleaved, right? So it would just avoid it completely. So now it's resistant, right? No matter how much of the drug you add, this enzyme will continue to cleave the ring. Every time it no longer becomes effective. Resistance. Okay. And then they can transfer this gene from cell to cell, from different types of bacteria, strain to strain, through different mechanisms, right? I'm talking about that. Transformation, conjugation, transduction. Okay. And the second one, it's related to modification. What do I mean by that? I mean something in the cell structure changes. So what would change, if you were to guess? Exactly, and that's what happens. Instead of having dealanine, sometimes bacteria will have dealanine serine. So there you go. Serine doesn't have a structure that looks like that these enzymes recognize. So what happens is they can produce another penicillin binding protein that recognizes the serine. And so now those enzymes prefer that combination to form the cross bridges, but it doesn't, those structures don't look like penicillin beta-lactam rings. So therefore the penicillin binding protein has no obstacle in front of it in terms of trying to decide where to go, right? Because there's only one serine and that's what it prefers. The drug doesn't look like that at all, right? So there's not a problem there. So now resistance has developed. Does that make sense? Okay, yes?
So, just to clear up, so penicillin binds to penicillin binding protein, correct?
No.
No. So binds to the dealanine.
Well, you're right. Penicillin binds to penicillin binding
Okay,
protein. Yes.
okay, cool.
Yes.
And so that's like we were saying of the paths diverges for the penicillin binding protein to either bind to the dealanine, or the penicillin, correct? Okay.
There's a competition. There's a lot of ways to explain it. It's like an analog, if you know what analogs are, right? It's a structure that looks like the native structure, right? So, however but you'll see the word suicide. And the reason they say suicide is because the process of the enzyme's completion never happens. And what that means is the completion of the enzyme's function is to bind and form a bridge, right? Whereas if it binds the penicillin, it'll bind perfectly, like it's supposed to, but then there's no product. So it's a suicide, the way they describe it in the literature, and you hear it's a suicide process, right? The enzyme binds to it, and there it is. There's no cross bridge. It just comes to an end, which is not good for the bacteria. But the enzyme's mind is, yeah, I'm still binding to something, and I'm doing what I need to do, but it doesn't really occur.
When penicillin binding protein is, or saying the dealanine, that's saying dealanine of one peptide versus a peptide creating a cross bridge to another dealanine, correct? Or is
Yeah, it's usually gap.
Okay.
Third one. So it's dealanine to a DAP, third amino acid. So when a penicillin binding protein binds to the two terminal alanines,
Yeah.
it releases one. That enzyme helps form that cross bridge, which forms with the adjacent stem peptide, which is the third one. So it's a 3-4. So it's alanine to DAP.
Okay. So
And the
why is it,
penicillin binding protein is what allows all that to happen.
so why is it dealanine twice?
Because that's the structure that the two the structure of the amino acids, that combination is ideal for the penicillin to bind. Okay, and those structures that happen to look like the beta-lactam ring play a key role. Okay, so it does require those two alanines that have that similar beta-lactam ring structure. It requires that to have a really good fit, like a perfect fit.
And those are the two at the terminal.
Terminal end, exactly. So
So
if you lose one, it may bind but not as well. Or if it's a serine, it may not bind and not bind as well. Remember, we talked about affinity, nothing's all or nothing. So when resistance happens, what that means is if it's a serine, it just doesn't bind as well. It doesn't mean it doesn't bind at absolute zero. It just means it's less binding, therefore we call that resistance. That's how everything works. So nothing's all or nothing. You know, we tend to think like that. Say, well, okay, it's now a serine, so it's absolutely does bind on. Well, not really. Just reduce the affinity so much that it appears to be non-binding, right? But the outcome is it's not effective. So, does that help?
So in the stem peptide, like we have 5 amino acids. The third one is normally the DAP, and then the last 2 are DAP and DAP, correct? Okay.
Yep. And then release one, you know, so on and so forth.
Okay.
So resistance happens, and then I do want to make a couple notes on this. Now we think about this. The transition that I want to make now is that, okay, we described this one of the very first ways of preventing growth, right? You have all that. So now the problem is resistance is so prevalent that scientists, you have to find another way to prevent growth, right? How do you do that? That's why we have this growth lecture, and that's where we have the growth research papers. As you think about it, so, well, how are you going to do this? Well, you need to find another way in which we can prevent growth. How do you do that? Well, if you explore and begin to look at specific details of how cell growth occurs, maybe there might be something in there that allows you to develop a new drug. And so that's where we begin to explore and identify that there's a lot of complex proteins called MREB, FTSZ, that are involved in cell division. Okay? So the thought is, as we discover these proteins and other proteins involved in cell division that are supporting segregation and septation, maybe those would be really perfect targets for drugs in the future that we could then use to prevent cell growth, right? So that's why they're so important. So now the amount of detail that we have related to that is now becoming quite evident, not only in the textbook, but in the research articles that we talk about. So that's the transition you have to think about, and it'll be obvious in the exam. And so you need to describe for me exactly what those proteins are. We've talked about 2 of them, okay, and how they support cell growth. Why? Because they might be future targets for drugs that we could use. Okay. And the last piece would be, what's the latest information that we have about the topics that we've covered so far? Well, if you think about your classmates' presentation, the MRB paper really talked about how they discovered that those proteins work together, and the fact that they talk about how they're tethered. That's tethering right there. How that is really important, meaning that it explains how you can have proteins that are positioned in the correct area of the cell, supporting peptidoglycan synthesis and making it successful, right? That's all in the introduction of that research article. Read it. And the last piece would be, okay, what's the outcome of some really interesting growth? Biofilms, right? That was popular. We had tons of questions about that. So tell me what biofilms are. Read that introduction article. It talks about how this coffee plant might be an interesting way of preventing that formation, right? So there you go. That's the exam, guys. So be ready.